California blues, dreaming all alone, nothing else to do.
Hello everyone. Nothing better than Roy Orbison to start a talk about a conference that was held in California. I had the opportunity and the privilege of attending the Plant Animal Genome Conference, which was impressive. I knew there was a big conference, but not that big. With that impressive trade show, is unbelievable what's the technology that's available nowadays to do genetic analysis in plant and animals. I also had the opportunity to meet some friends, which include Dr. Miller, who works for the American Young Association, at your right in the screen, and also Dr. Mehdi Salagozé, who works for Semen Alliance in Canada. My talk was invited to talk, which is always great when people invite you to give a presentation, and even more exciting when at the end of the section, the chairman comes to you, shakes your hand and say, "Oh, please make sure you contact me for a contribution for next year." So that's great. I think that what you present reach their standards of quality, and you manage to keep the audience awake, which is always a plus. So in that section we have a lot of contributions. Most of them are related to plants, so it was always a challenge when you are going to talk about bull and cattle in that context, but I think that we managed to address our points. The contribution that I provide in this conference is linked to a publication that we got published last year in the Reproductive Biology Journal. So I should also acknowledge Dr. Mariana Dial de Morin, who is at Cornell University, Dr. Rob Foster and Dr. Schneider, who are both at the University of Guelph. So the talk was entitled Seminal Plasma Proteomics Vary with Feed Efficiency and Fertility-Related Measures in the Bovine. And starting with a brief introduction, we know that in the way that we used to select sires was more like based on a visual appraisal. Was like walk around the animal and checking, oh, it has a nice rump, it has a nice chest, has a nice leg, so that should be a sire. And we know that we have been refining that over the years, although the visual appraisal is still reality in the beef cattle industry, that's for sure. So we still need to put some more work on that. And my niche in science has been to study phenotypes. That's what I really like to do, and it's really studying biology, physiology, and how I could be pleasing a geneticist, how I could be pleasing somebody that works strictly with nutrition. So it's really like being on the bridge and trying to find new phenotypes for complex traits. The main areas of interest have been feed efficiency, and also fertility and health traits. So those are the main pillars of a complex trait that I have been exploring, and have been doing that in a very diverse manner. We have been studying the behavior of the animals, we have been looking at organ cell function, the heat balance through many different techniques, and also evaluation of the breath gases and body fluids. So today our talk is within the agenda of body fluids, and we're really here focused today to talk about semen. So when it comes to sexual development and reproductivity, we have been observing antagonistic relationship. We have observed that as we improve production, we decrease reproduction in a variety of species, include swine, turkeys, and also poultry, dairy cows, and a lot of evidences are being released over the last few years in the beef cattle. So our lab has been put a lot of emphasis on the study of sexual development and fertility of young bulls, which is like of primary interest for the industry, because the sexual development of the bull is quite associated to what will be the fertility of the females, and also for the whole impact the whole generation interval of the species. And what we have been observing, that in general, as we improve feed efficiency, you have building up a decrease on sexual development and semen quality. So these are some of the phenotypes here that we have been looking at. So that includes organ biometry, seminal tubulus histology, testis ultrasonography, sperm quality, and also infrared imaging. Also, there is a lot of evidence supporting association of seminal plasma proteins with fertility. These are just a few examples of publications in key journals highlighting that, and if you search you find more and more. So that's like well established, the association of seminal plasma proteins profile and the fertility of the ball. Thus, our objectives in this study was really to study the potential relationships of seminal plasma proteome with age, with production traits include feed efficiency and carcass compositions, as well as a long list of fertility-related traits as I just introduced. Move on to materials and methods. In this study, we have access to a population of 31 crossbred bulls. Bulls are semen sampled about 1 year of age, and the breeding composition of the bulls was mainly Angus and Simmental. These bulls were fed a ration based on high moisture corn and also alfalfa. And as part of the performance test, it was evaluated the individual feed intake, ultrasound for body composition, and also body weight. So at the center here we have the timeline of the experiment. The animals are subject of feeding performance for 112 days, and then a couple weeks later did a breeding soundness evaluation, and a couple weeks later we slaughtered the animals. At slaughter, we assess a few different traits, which include carcass weight and also carcass composition. So for the carcass composition, you use the rib dissection technique for that. So which is like established from a long time ago. So we got a section of the ribs, of the loin and ribs, and then we dissect that into lean, fat, and bone, and with that you could infer about the whole body composition. For feed efficiency, we based our calculations on residual feed intake, which was given by a linear regression on body weight, feed intake on body weight, vegetative gain, back fat, rump fat, and ribeye. You see that we include here ultrasound traits, and those are quite important to accommodate variability in body composition. So we have back fat and rump to account for fatness, and ribeye area to account for leanness. Just a quick explanation about residual feed intake. Basically, with this trait, the more energy and nutrients that the animal deviates towards production, towards growth, the better will be the efficiency of the animal. While the animal that has a higher maintenance, a more costly accretion of body tissue, that animals become inefficient. When it comes to fertility-related traits, one of the first assessments that we did was the testicular ultrasonography. This is a very promising assessment. It takes you 30 seconds or less to do each testicle, and with that you have a very good snapshot of the sexual development of the bull. The image that we collect, we use an image analysis software to address the pixel intensity on that area. And we know that animals that are more sexually developed, they have more structure, have more microtubulus, they have more protein in the testicle, which gives a higher pixel intensity. So that's pretty much how this association works. Another assessment that we did was to take infrared thermographs from the rear end of the animal, focus on the scrotum, and on those images we look at the temperature variability at the base and also at the tip of the scrotum. Organ biometry was also another target of our research. So we start with the scrotal circumference, which is by far the most common trait that's applied in the selection of bulls in the beef industry, and this has been incorporated for a long time ago. And since these bulls are slaughtered, we also could measure some organs like more directly, such as the testis weight and also the epididymis weight. And with the testis, we could do some histology on that tissue, where we evaluate the maturity of the organ by looking at this seminiferous tubulus. So you could check the proportion of a seminiferous tubulus that are classified as immature and the ones that are classified as mature, based on the cellularity, based on the structure of those microtubules. We collected semen in this study using electroejaculation, and the semen was immediately used for evaluate sperm motility. We're particularly interested in the progressive sperm motility, which is basically addressing here the percentage of good swimmers in that semen sample, and for that you use the CASA system from Minitube. We also look at the sperm morphology. So we checked the proportion of sperm cells that had head defect, mid-piece defect, loose head, tail defect, among others. So these are just a few examples of that. And as for seminal plasma proteomics, so after electroejaculation, a subsample of the semen was centrifuged. That was centrifuged twice. At 2 different speeds to ensure that the seminal plasma is clean of sperm cells, and then that sample was protein normalized to 1.5 milligrams and submitted to a mass spectrometry. The work was done at the Sick Kids Hospital in Toronto, where they have a world-class proteomics center called the Spark Biocenter, where it can do a lot of this proteomic analysis for us. As a result of this proteomic analysis, we got a long list of proteins, like a never-ending list, and from those we observed that 176 have a normal distribution. And since we are dealing with a small population, we found wiser at this stage to be focused, to be narrowed down to the proteins that have naturally a normal distribution. And for those 176, we went back to the scientific literature, and then we separated 17 of them that are recognized to be fertility-associated proteins. So to really focus on the ones that are previously described by other authors. As for statistical analysis, we start with a categorical analysis. So here we try to work in a similar way as we do in reality in the like in commercial herds for selecting bulls. We know that from a group of bulls, a very small percentage of bulls will actually become sires. So what we did here, from a population of 31 bulls, we picked the 7 most and the 7 least efficient bulls for doing a protein comparison analysis among them, like compare the traits among them. And from the 17 proteins that we elected, we did a protein principal component analysis on that and extract 3 main factors 1, 2, and 3. And then we did a series of correlations of factors 1, 2, and 3, and also with each individual protein, with the reproduction-related traits that we assess, performance traits, and also age. Alright, so move on to results and discussions. The first set of results that I have to share here was the comparison, the category comparison between efficient and inefficient bulls. And for this, I separate some proteins that have very interesting results, very relevant to be discussed. The first one was the epididymal sperm binding protein, where we observe a higher concentration of that protein in the efficient bulls. This protein is associated with dead sperm, so the more dead sperms you have, the more you have that protein, and which makes sense because from our studies we have observed that efficient bulls, they have a higher percentage of dead sperms. Also, we found higher concentrations of the Semen peak type C2 protein on the efficient bulls, and that protein is a precursor of the first protein that I just described, so that makes complete sense. A third protein to discuss here is the glutathione peroxidase. So this is a protein that has antioxidant properties, and because animals that are efficient, they have a lot of structural problems with the sperm and dead sperms and so on, you end up having a high prevalence of reactive oxygen species, so that higher concentration of GPX might be alternative from the organism of the bull to deal with that increased concentration of ROS. And the last protein to talk here is the concentration of protein C inhibitor, which observe a higher concentration in the inefficient bulls. and that protein is actually positively associated with sperm quality. So the better concentration, the higher the concentration of that, more likely the better be the semen quality of that bowl. So the bottom line of this is really indicating us that in many different ways, our efficient bowls, they have a feature of delayed sexual maturity or a potential delayed lower fertility based on the seminal proteomics profile. All right, so move on to some correlations data that I want to show to you. In this slide, you have the correlations between age and body size with a list of proteins. It's interesting to observe that these 5 proteins in particular, all of them, they have negative correlations with those traits, and also correlations of a similar magnitude, and that really is probably associated to the fact that age and body size are associated in the bowls. And there's a lot of discussion around this proteins here. I keep myself short, but when the time that we sample these bowls are where they are about 1 year old, and that's a trick time for evaluating the sexual development of the bowl, especially because a lot of these traits are associated here with testosterone. And testosterone is like on the turning point, is like starting to decrease the concentration after the pubertal period. Another point to bring up here was the although not presenting numbers, we observe positive relationships with key proteins for sexual development and fatness, and negative associations with leanness. So that makes sense because we know how important is the fatness and the leanness to the sexual development of the bowl. So those are important facts as well. When it comes to seminal proteins and organs biometry associations, you could observe positive associations with phosphoglycerate kinase 2, all positive associations, and that protein is particularly important for the reproductive tract development. There are studies showing a positive association between the number of germ cells in the testicles, seminiferous tubules in the testicles, and the concentration of phosphoglycerate kinase 2. And also, to keep the story short here again, the spermidine had a negative association with all those, and that's why it's also interesting, because one nickname for spermidine Z13 is also the anti-fertility factor. So that makes sense to see that those negative associations here. Moving on, when you look at the correlations between histology and seminal plasma proteins, you could observe a positive correlation between the prevalence of mature tubules with cathepsin and seminal plasma protein 1. And that makes sense because these two proteins are related to immaturity, sexual immaturity. On the other hand, we observe positive correlations between the prevalence of mature tubules with glutathione peroxidase and osteopontin, which are two proteins directly associated with sexual development. When it comes to semen quality, we found positive correlations between progressive motility of the sperm cells with a seminal plasma protein 1 and acidic seminal fluid protein. These correlations are interesting, and this is where phenotyping can be a challenge sometimes, or can be a bit tricky. So these correlations are positive, although some people could observe a negative association with those. So what's going on here? It's a matter of the buffer that we use. In our research, we did evaluate our seminal plasma using a buffer that has tris, DTA, and egg yolk. That buffer, it is known to affect the energy used in the sperm cell. Basically, those two proteins here, they're bounded to the sperm cells, and they're there making ensuring that the sperm cells are saving energy. And just like a detachment, it's really time to swim towards the egg. So when we did that in our study, those proteins were detached from the cell right there, and then when we did the motility assessment, we had a high motility on that. So we need to be just a bit careful what type of buffer and what's the conditions that the trait is actually being assessed. And two last sets of correlations with fertility-related traits are presented in this slide. The first one are the positive correlations between the variability of the temperature in the base and tip of the scrotum with acrosine and phosphoglycerate kinase 2. Those two proteins are associated with the mitigation of reactive oxygen species in the seminal plasma. And when you have a mitigation of ROS, means that oxygen is being consumed, so energy is being released. So that has an impact of the thermoregulation and the radiating heat loss of the scrotum. So that might be our first shot on this. We need to further work on this and see if we're still getting consistent results in this way. But certainly the possibility of assessing the fertility capacity or reproduction capacity of a bull through an infrared imaging analysis, that can be, it is quite exciting, and we need to do more work on that. And still related to imaging, we had positive relationships with seminal plasma protein 1 and spermidine Z13. And this is interesting because the more protein you have in our organ, in our case in the testicle, the higher will be the echogenicity of that organ. So that positive correlation here with seminal plasma protein, which is one of the main seminal plasma proteins in the bowel, it is quite interesting and remarkable. So move on to the last set of results. We have the breakdown of the variability of the 17 plasma proteins according to the principal component analysis, and you could observe that factor 1 and 2 and 3 together, they explain over 70% of the variability, with factor 1 and 2 accounting for about 60%. And we did some correlations between those factors with the different groups of traits that you observe, and was quite interesting to observe that three main classes of traits are the main responsible, the key associations with the variability in the seminal plasma proteins, which include body size and composition, and then age, and then organ biometry. With this, I'd like to wrap this up with some conclusions. The first one is that expression of fertility-associated proteins vary according to feed efficiency, and you could observe that based on the profile of proteins of bowls, of comparing efficient to inefficient bowls, is that the efficient bowls may have a delayed sexual maturity based on that seminal profile, plus based on the long list of evidence that have been building over the years on this regard. Also, that the expression of fertility-associated proteins is linked with sexual maturity in young bulls. So that was well addressed with the different correlations that we have with different classes of traits. And also I'd like to do some acknowledgements. First, to acknowledge the government agencies that have been supporting these and other studies from our research group. That's very valuable. Same is true for the substantial support that have been getting from the industry over the years. Without the financial support, we couldn't do any of this job. So we really thank you all that support. And with that, I'd just like to wrap up this presentation saying that we have more work going on the bull fertility and proteomics. Actually, now we are working a population of 50 bulls, so it's a larger population, where we had the conditions of doing proteomics on the seminal plasma. We did metabolomics of the seminal plasma. We did a lot of the organ biometry, like you're moving to a higher level. That's like talk about 7 or 8 different organs now, all related to the reproductive function. And also we have the genomic information of those bulls. So we have more conditions to spend our research on this, and hopefully we end up within a situation that we can identify a handful or less than that of key parameters that we can be incorporating in the practice of evaluating sexual development in bulls, which is key, which is important, and we need to face this and deliver solutions that will make impact to the industry. Also, I'd like to tell you that this and other presentations and my full scientific profile, it is available on ResearchGate, so you can find every single publication that I ever did in there. So you're welcome to visit my profile. And if you are watching these videos, that's because you're not familiar with our channel about Taurus efficiency. It takes a lot of work to put this together, and it would be very nice to have, if you are benefited from this presentation, so it would be very nice to have you become a subscriber from our channel. This is all focused on science though, so it is really be beef cattle, dairy cattle, and sheep production, maybe goat, and be all updated in that way to benefit research, extension, and training. Thank you, and see you next time.